the plant also. Okay, so and it is like very widely present, or we can say almost most of the body tissue are producing this histamine. And this histamine is mostly we are able to see in the pathological condition like the hypersensitivity, even like the tissue injury. Whenever tissue injury occurs, the injured cell will going to release the histamine, okay, in that area. That you all are aware about the inflammation process as well as hypersensitivity, mostly type 1 type of hypersensitivity, in which mast cell will going to release the histamine, okay, and they will going to carry out the redness, itching, as well as swelling also in that area. So that is about the pathophysiological, but physiologically they are also present at various locations. Okay, so we're going to talk about which locations are that, where they exactly present, and what kind of physiological role they are playing in our body. So if we're going to see in our body, we'll have two types of histamine. One is called as mast cell histamine, and second one is called as a non-mast cell histamine. Okay, means what histamine which is present inside the mast cell, and second one the histamine which is not inside the mast cell, but they are present, uh, they are synthesized by that cell particular. Okay, and they are not stored into the granules, they are synthesized by that cell, uh, that we're going to talk. So here you can see the central nervous system, which is a non mast cell kind of things. There are the neuron for that. Okay, similarly if you're going to see the stomach, fundus part of the stomach, there also you will able to see non-mass kind of non-mass cells are synthesizing the histamine. So the mainly, if you will able to see the histamine location, the primary site are this one: skin, intestine, even the gastric mucosa, lungs, liver, placenta. These are the primary site for this such a histamine. And if we're going to see apart from this site, other sites are also there, like central nervous system. Even these are present in the stomach, even they are present in the epidermis, gastric mucosa, and even the various growing regions like various epithelial cells, they are having this histamine present. Apart from that, it is present in the blood, body secretion, various venoms are also having this histamine, and pathological fluids, like whenever there is an inflammation kind of things, in that also you will able to see the histamine. Okay, so now we're going to talk ki how they are, how our body is synthesizing the histamine. So our body is synthesizing the histamine in this way. Like you can able to see the histidine, the amino acid. This is responsible for synthesis of histamine. So the enzyme which is present, like histidine decarboxylase, is very important to convert this histidine into the histamine. Okay, so they are causing the decarboxylation. You can see here carbon dioxide get removed from the histidine and they are forming the histamine. Okay, so after they are forming in our body, this histamine will going to work, like they will going to, how they will going to work, that will going to talk later. But how they get removed out from our body, that will going to see here. And then we're going to see key in between that what happens, means after synthesis how it will going to act and how it will get removed. So to remove out from our body, we are having these two things in our body, like these are mostly present in the liver. Okay, so even they are also present locally also, not only the liver. After their work, it get destroyed. Okay, and if they enters into the systemic circulation, liver will going to uh, carry out their metabolism. So here histamine, how they get degraded, how they get metabolized, they are metabolized by these two particular enzymes. The name of that enzyme is histamine and methyl transferase. Okay, and methyl transferase. So name of that enzyme is here. Here DAO, that stand for diamine oxidase. Okay, so here transferase enzyme is here, and one side oxidase enzyme is here. You can see here. So these are the metabolized by cytochrome 450 system. Okay, that is mostly having these two enzymes, N-methyl transferase as well as the D-amine oxidase. Okay, and after that it get eliminated into the urine. So two type of reaction occurs. One is called as a methylation, and second one is oxidative deamination, by which they get converted into the imidazole acetic acid, and they are removed out from our body in the urine. Okay, so this is about the synthesis, how they get metabolized. Now we're going to see about the storage. Okay, how they are stored in our body. So where they are present in the mast cell. So mast cells are storing them. So how they are storing them? They are storing into the granules. Okay, so what this granule consists of? How this histamine present in that granules? So this, in that granules you will able to see the histamine along with some proteins. Like you can see the protein which is called as a acidic protein and heparin. Okay, these are the two protein along with this histamine present. I will show you the diagram. Just a second. I guess it is, I missed that diagram. Yeah, here you can see. So these are the mast cells. In that mast cells, such a granules are there. So inside that granules, histamine is present. But histamine is not present alone. They are present with some protein like acidic protein as well as heparin, and both these are the negatively charged and our histamine is positively charged. So because of that they are binded with each other, positive negative charge. Okay, so whenever they will get released, again that histamine will get separated out from these proteins, and then this free histamine will going to work on the target, or we can say receptors, and after that we can able to see the action of the histamine. Okay, so in this way this histamine uh, get stored in the mast cell. Okay, held by the acidic protein heparin, and histamine is positively charged. And if we're going to take the histamine orally, it will not going to work. Okay, if we'll try to take histamine in the form of means from the intestine, okay, the intestinal cell will going to sorry, liver will going to destroy it, not intestine. So because of that it will ineffective if they are given orally. Okay, so this is about the what we had seen, biosynthesis, we had seen about the metabolism, we had seen about the storage. Okay, so now we're going to see the very important thing. This we already seen where they are present, we already seen the distribution too. Okay, so how they will going to act? They will going to act via the receptors. Okay, so means synthesis, after synthesis they get enter into the mast cell. Once they release from the mast cell after the stimulation, they will enter into the uh, surrounding area, and they are having the receptor to act. So what kind of receptors they are having? They are having the receptor like H1 receptor, H2 receptor, H3 receptor, and H4 receptor. These are very specific for histamine. Okay, and the target cell, if they are having such a receptors, then we are able to see the action of histamine on that cell. So what kind of receptor they are? They are G protein types of receptor. G protein couple type of receptor. All these four receptors are of the G protein couple type of receptor. Okay, so here, but they are having the different mechanism. As you know, G protein also have the different types of mechanism, like you know about IP3 DAG kind of mechanism, like the phospholipase C get activated, which will going to convert into the IP3 and diacylglycerol, and then it will going to activate the protein kinases, and that protein kinases will going to show their effect. Okay, here you can able to see adenylate cyclase. Okay, the adenylate cyclase will going to form the CMP, and that increase in the CMP will cause the activation of further protein kinases. Okay, so this mechanism, how they are working via the CMP pathway, how they are working IP3 DAG pathway. Okay, and H3, they are like the inhibitory kind of things. They are mostly present at the post ganglionic part. If you're going to see the effect of H3 receptor, wherever the H3 receptors are present, what they are doing, they are inhibiting the release of histamine. Okay, they are called as a post ganglionic receptors. Okay, and they will going to decrease the release of the histamine at that particular position. We're going to talk in detail later on. Just you need to see which are the different receptors are there. H1, H2, H3 receptors are there. Which type of receptor it is? G protein couple type of receptor. How H1 acts? H1 acts via the IP3 DAG mechanism. Okay, how H2 is act by the adenylate cyclase or CMP mechanism. How H3 is act by acting on the ion channels. Like you can see the decrease calcium influx as well as potassium channel inactivation. Okay, so these three are, you can able to see only three are here, but actual there are the four receptor, H4 receptor. But this H4 receptor is not still fully explored. It recently discovered around like 2000, 2002. In that year it got discovered, and later on found out that this H4 is responsible for various allergic reaction. Okay, and we are scientists are trying to find out the what is the shape of this receptor, what type of receptor, where it exactly present. So these H4 receptors, we are able to see some location of the H4 receptors, but still we are not able to get any kind of potential drug to be acted on these H4 receptors to stop the allergic reaction. So whatever the allergic drugs are available, they are acting on this H1 receptor and H3 receptors. Sorry, mainly the H1 receptor antagonists are available.